Hi guys, welcome back to my channel. The course at hand is Cytogenetics, and for this topic, we'll be talking about the central dogma. If you're ready, then let's start. When we talk about the central dogma, this was proposed by Watson and Crick in the year 1950. So immediately after they discovered the double helix model of the DNA, they then proposed the central dogma. And according to these two innovators, they described the central dogma as the relationship between nucleic acids and proteins, and they described the directional flow of information. They even said that the central dogma deals with the detailed residue by residue transfer of sequential information. And this image right here perfectly sums up that description that they gave. So according to them, in order for protein to be produced, DNA should first be duplicated into a copy called RNA, and the RNA will be the one to code the protein. So basically, they were able to include into detail or put into detail the transfer of information from DNA to RNA to the eventual production of protein. So the central dogma is actually composed of three important steps. These steps are transcription, translation, and DNA replication. And in order for us to better understand the process or the flow of information in the central dogma, we have to have a good understanding of the DNA structure. So we are going to talk about this first, no? What is the DNA made up of? How are they made before we go into the central dogma proper? So DNA are composed of nucleotides. These are the monomer units or the building blocks of nucleic acids, both for DNA and RNA. So basically, your nucleotides are composed of a sugar, a nitrogenous base, and a phosphate group. So please do remember that your nucleotides are composed of your sugar attached to your nitrogenous base and attached to a phosphate group. These nucleotides, aside from functioning as the building blocks of both DNA and RNA, also have other functions inside the body. First, they function as coenzymes. We know that enzymes, no, ang trabaho ng enzymes is to break down certain substances. And sometimes these enzymes would require other substances in order for it to be able to break down what it needs to break down. And those substances needed by the enzyme are called coenzymes. And these nucleotides, no, the triple combination of phosphate, sugar, and nitrogenous base sometimes act as a coenzyme. Secondly, they serve as donors of the phosphoric group, specifically for ATP and GDP, of sugars and of lipids. So later on, we'll discover that these nucleotides, kamag-anak pala yan ng mga ATP, GTP mo, no? What is ATP again? This is the commodity of energy used by the body, no? Your adenosine triphosphate. So later, we'll go to a slide highlighting yung relationship of your nucleotides to your ATPs, GTPs, etc. Also, when you study your biochemistry, you'll find out that nucleotides also function as regulatory second messengers. And lastly, they also are important or used for chemotherapy and immune suppression. Okay? So again, we'll be talking about the different components of your nucleotides since the nucleotides are the ones that create or build your nucleic acids. And we'll start the discussion with the nitrogenous bases. All right? So nitrogenous bases are nitrogen-containing heterocycles. They can be a single ring or they can be a double ring. But what is important is that aside from your carbons, no, they also contain your nitrogens, no? Let me just remove myself from this view. There you go. Yeah. You see your nitrogens, no? Both in the single ring and even in the double ring structures of our heterocycles. So, the cyclic compounds other than your nitrogen also contain carbon and other elements. And But the most important part about nitrogenous bases, they are the information containing parts of the DNA. Not the sugar, not the phosphate groups, but the nitrogenous bases. Okay, they are the ones that carry the information. And we have to remember that DNA is measured in the number of base pairs. No, I'm sure you've already heard from me in the past that DNAs are very big structures. No, that is why they have to be condensed in a chromosome kasi napakahaba ng DNA. And DNA is usually described in terms of how many base pairs they have. So when they have 1,000 base pairs, they are described as kilobase. And when they have 1,000,000, they are described as megabase. Now, it is important for us to know that there are two types of nitrogenous bases. These are your purines and pyrimidines. Now, what is the difference between the two? Your pyrimidines are 6-membered rings containing 2 nitrogens. These are the ones on your right, no? 6-membered rings, 1, 2, 3, 4, 5, 6, and 2 of those are made up of your nitrogen. There are 3 types of pyrimidines which are involved in our nucleic acids, and these are cytosine, uracil, and thymine. Please forgive me, this should be thymine, okay? And then your purines, which are 9-membered rings forming 2 rings. Forming 2 rings, as you can see in this image. Now let's count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9-membered rings composed of 2 rings. All right, and we have 2 types: your adenine and guanine. Again, these nitrogenous bases are the ones that carry information and connect to your sugars and phosphates to form your nucleotides. So, these base pairs or these nitrogenous bases complement each other, meaning they form pairs, no? They form partners. Your adenine is partnered with your thymine, while your guanine is partnered with your cytosine. Notice that in the DNA, we do not use uracil. Okay? Instead, we use thymine. But for RNA, we use uracil instead of your thymine. Now, what is the importance of these complementary base pairs? No? First, it is the partnership between the single ring pyrimidine with a double ring purine. So it's always like that, guys, ha? It's always a pyrimidine partnered with a purine. Never a purine with a purine or a pyrimidine with another pyrimidine. It's always that single and double ring partnered together. In this continuous cycle, this continuous chain of double ring plus single ring, double ring plus single ring, forms a symmetrical double helix formation. Kaya, very smooth, very symmetrical, um, ang formation ng ano natin, DNA natin kasi na-maintain nyo yung double single formation niya. Imagine if you have 2 double rings, edi hindi magiging symmetrical, no? Magkakaroon ng extensions in the formation of your DNA. So, please do remember that your base pairs are connected by what we call hydrogen bonds. Okay? So, in this discussion, we'll also be talking about the bonds that hold the different parts together. So, in between two nitrogenous bases, they are combined by your hydrogen bonds. According to our book, Lewis, hydrogen bonds daw are weak bonds. That's why they are easily opened by your helicase or your RNA polymerase. So that's it for our nitrogenous bases. Next, we have your sugars. And the sugars that are being used for the nucleic acids fall under the classification of pentoses. Pentoses are 5-membered rings of sugar. So let's count them up. 1, 2, 3, 4, 5. So 5 sides forming your 5-membered rings sugar. There are different sugars used for DNA and RNA. For RNA, we have ribose. And then for DNA, we have your deoxyribose. So what is the difference between the two? The attachment on the second carbon. All right, that's the difference between the two. For ribose, we have a hydroxyl group attached to your second carbon, while for DNA, we have 2-deoxyribose because the oxygen in the hydroxyl group has been removed. So, deoxyribose na po siya. No oxygen in that area. And when you add your sugars and nitrogenous bases together, you will get your nucleoside. Okay? So again, nucleoside are the combination of your purine and pyrimidine attached to a sugar. And your sugar and nitrogenous bases are attached by BN glycosidic bond. BN glycosidic bonds. So for our complementary base pairs, they're connected by your hydrogen bonds. But when we attach the sugar to the nitrogenous base, we have to use a BN glycosidic bond. I also want you to notice that when we talk about nucleosides, they change their names, no? From adenine, cytosine, guanine, and uracil, nag-iiba yung pangalan nila. Then, now we have your adenosine, we have your cytidine, guanosine, and uridine. Okay? So, hindi na siya pareho nung name nung kanilang nitrogenous base pair. Once you attach a sugar, they change their names. Okay? And of course, the last part of the DNA is your phosphate, no? So this phosphate forms the backbone of the DNA. And as mentioned, it runs, itong dalawang strand na to, magkaiba yung direction nila. One strand runs from 5' papunta ng 3', while the other strand runs from 3' towards your 5' end. Alright? And this opposite running direction is called anti-parallelism. Okay, it's called anti-parallelism. And the the phosphate is attached to your sugar by an ester bond attached to the hydroxyl group of the sugar. Okay, so again, the phosphate is attached to the sugar by an ester bond, specifically to the hydroxyl group of the sugar. So let's try to review the bonds again, no? Complementary base pairs are attached to each other by your hydrogen bond, while your sugar is attached to your nitrogenous base by your BN glycosidic bond. And of course, your phosphate is attached to your sugar by an ester bond, specifically to the hydroxyl group of sugar. So those are your nitrogenous bases, your sugars, and your phosphate group, forming your nucleotides. And again, nucleotides are the building block monomer units of our protein. Sorry, of your DNA rather. So these are the base formula and then the way that they change their name once we add the sugar and the phosphates. So the base pairs are adenine, guanine, cytosine, uracil, and thymine. Once you add the sugar to it, it changes its name from adenine to adenosine, guanine to guanosine, cytosine to cytidine, uracil to uridine, and thymine to thymidine. And then once we add the phosphate, it changes name again. Adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, uridine monophosphate, and thymidine monophosphate. So now we were able to understand now, no, yung yung relation niya doon sa function kanina of being a phosphate donor. No? Kasi once you have your AMP and you add 2 more phosphates to that, that becomes your adenosine triphosphate. Okay? And your adenosine triphosphate will have to donate their phosphate in order to produce energy. So putting everything together, this is how they would look like, no? You have your phosphate backbone attached to your sugar via an ester band attached to the hydroxyl group. Your sugar is attached to the base pair via BN glycosidic bonds. And then your base pairs are attached to each other by your hydrogen bonds. The combination of phosphate, sugar, and nitrogen base forms your nucleoside, while the combination of sugar and base pair forms your nucleoside. Okay? Now, the greater curvature formed by the double helix is called major groove, while the smaller curve is called minor groove. Before we go into DNA synthesis, I would like first to tell you that once we have an original parent and it undergoes DNA replication, no, the daughter, the daughter DNA will both have one strand of the parent. Okay? Tag-isa sila ng parent strand nila. Right? But after the second generation, only one out of the two daughter cells will have that original parent strand. No? The other one will be completely new na po. Okay? So this means that sometimes the original trait from the parent is not carried over to the offspring because of this formation of new generation DNA molecules. And this is the reason why we have your dominant and recessive alleles. So now let's go to the central dogma proper. We'll talk about DNA synthesis or replication first, no? And when we say DNA synthesis, it is the production of new DNA essential for cell division. And this happens in what phase of the cell cycle? S phase, okay? So DNA synthesis or DNA replication occurs in the S phase. So you have to remember that DNA synthesis always proceeds in the 5' to 3' direction. That is why one RNA strand will be formed not pababa while the other one will be formed pataas. Kasi yung isang strand natin 3' to 5' while the other one 5' to 3'. So in order for it to follow the 5' to 3' direction, they will have to go opposite each other. But there are requirements needed in order for DNA synthesis to occur. These are, first, we should have a free 3' hydroxyl group. We should create two single-stranded DNA as templates. And the process of DNA synthesis is a semi-conservative process. Why semi-conservative? Because each DNA double helix will be able to conserve half of that original parent strand. Okay? Dahil may naiiwan pa na part ng parent, we call it semi-conservative process. So this is the overview of DNA replication. Ano bang nangyayari sa entire process of DNA replication? First, we start with the parent DNA molecule, no? So, we will choose what DNA we want to duplicate. And then the parental strand will unwind and will separate at several points. Each parental strand will then provide a template, no? So, one parent strand, itong single-stranded DNA, magiging template siya. And the other one as well, magiging template siya for the new DNA strand that we will produce. And then, they will add nitrogenous bases to each nitrogenous base that is exposed. Eventually, after the production of your new DNA strand, the sugar phosphate backbones of the daughter strand will close. So this is just an overview. We'll go and study it in depth starting now. So the first step in DNA synthesis is the unwinding of DNA. I have here on top the different enzymes needed for DNA synthesis. So the first step is DNA helicase, okay? Helicase, tandaan yung letter H. Why? Because siya yung naghihiwalay. The helicase will bind to the ori protein of the parent strand and will start to separate the strands, okay? It is the helicase that naghihiwalay, that separates the original strand or the original parent DNA. And saan siya nagsa-start? Sa ori protein of the parent. So it will look for areas that are rich in A and T, your adenine and thymine base pairs, and those two strands that will be produced will be stabilized by your binding proteins. So this structure here, ito, tsaka ito, tsaka ito, those are your binding proteins which keeps the single-stranded DNA separated or set apart, no? Pinaghihiwalay sila. Yan. So what does that again? Your binding protein. What opens it again? Your helicase. And then lastly, another enzyme called primase. This violet oblong here, no? That's your primase. primase will make a short stretch of RNA on the DNA template following the 5' to 3' direction. So the primase just initiates the synthesis of an RNA molecule from the DNA template. Siya lang yung nag-i-initiate, siya lang yung nagsa-start, but it is not responsible for the elongation of this new RNA strand, which will eventually become your DNA. So step 2 is the formation of the replication fork. What is the replication fork? It is the appearance of the unwound DNA, especially when it starts to form na the leading and the lagging strand. So imagine, this is the handle of the fork, and then this is the one of the spokes, no? Another one. At pag humaba-haba na tong pinoproduce natin na continuous and discontinuous strand or leading and lagging, mas magmumukha na siyang tinidor, di ba? So that is your replication fork formation. What keeps it open? Your binding protein. Next, step 3 is DNA polymerase replication. So as mentioned, primase will just initiate the formation of your RNA strand, but it will be DNA polymerase, which adds nucleotides to the RNA primer. Basically speaking, the DNA polymerase is the one that elongates that RNA strand, which will eventually become part of your DNA. Not only does the DNA polymerase elongate the strand, but they also perform proofreading activity in order to replace mga incorrect bases. Galing, no? So, hindi lang niya pinapahaba, pero it also functions for proofreading. Okay? So, how does it do it? So, again, here's your parent DNA. You were able to separate the two strands, no? You have the leading strand and the lagging strand. And then, your DNA polymerase will be the one to add your nucleotide bases in the 5' to 3' direction. No? So, it performs the elongation of your DNA strand. Moreover, it adds nucleotides first doon sa ginawa ng primase natin na RNA primer, and then it grows the chain using phosphodiester bonds. Okay? Paano pinapahaba ang DNA? By putting them together via phosphodiester bonds. And of course, the most important function, the proofreading. So yung proofreading natin occurs by 3' to 5' direction. And pag may nakita siya na incorrect nucleotides, it will activate your exon nuclease to remove that incorrect nucleotide and replace it with the right one. Okay? So, ang nagpo-proofread, DNA polymerase. What removes the incorrect nucleotides? Your exonuclease. Step 4 is the formation of your Okazaki fragments. So, alam naman natin, di ba, na dalawa yung strand na napoproduce ng ating template DNA. One has continuous strand synthesis, no, again, following the 5' to 3' direction, while the other one has discontinuous production. The continuous one is called leading strand, while the discontinuous one is called the lagging strand. Each discontinued portion is called an Okazaki fragment. Okay? An Okazaki fragment. And eventually, there will be a removal of your RNA primers using your ligase. So si ligase po, what it will do, it will remove the RNA primers kapag kompleto na yung base pairs. Moreover, it will seal the sugar phosphate backbone. So, from hiwa-hiwalay na Okazaki fragments, it will connect them together in order for a new daughter DNA strand to be produced. So, what removes the incorrect nucleotides again? It is your exonuclease. But what fills in the gaps are your DNA polymerase and then isasara ng DNA ligase. So once the DNA has been reproduced or replicated during the S phase, it will then enter mitosis. And specifically, it needs to be condensed, no? or else, again, masyadong makapal yung chromatin. So the DNA molecules, being extremely long, needs to be packed into a chromosome. No? So from millions and thousands of base pairs long, it will become only 2 micrometers long. They shrink by a factor of 7,000 times. No? Grabe yung difference. 7,000 times smaller ang chromosomes compared to your DNA. And how are they able to do it? By DNA coiling around proteins called histones. Itong mga histones na to, ang mga DNA natin, once naka-wrap around the histones, they will resemble beads on a string appearance, and that combination of DNA and histone is called nucleosome. Okay? Nucleosome. Here are the enzymes I mentioned which are involved in DNA replication. Your helicase na naghihiwalay sa parent DNA mo, no? It unwinds the parental double helix. You have your binding proteins which stabilizes the two strands, no, or separates them apart. Your primase which initiates the synthesis of your RNA primer, no? And then your DNA polymerase, which is the one that adds nucleotides, elongates the new RNA strand, and even performs proofreading. And then finally, your ligase, which will be the one to join your Okazaki fragments and also seal the sugar phosphate backbone to the nucleotides attached by your DNA polymerase. So that is for your DNA synthesis or DNA replication. Next, let's talk about RNA. Basically, RNA is analogous to your DNA. Ibig sabihin, halos pareho lang sila ng structure, no? Magkamukha sila. They are composed of your nitrogen base attached to your sugar via a BN glycosidic bond, and then it's attached also to a phosphate backbone via an ester bond on the hydroxyl group of the sugar. So pareho lang, no? The difference is, instead of thymine, RNA uses uracil. Moreover, look at this, it's single-stranded. Single-stranded lang po ang RNA natin. So in order for us to summarize the differences between DNA and RNA, we have this slide. Ayan. So DNA is usually double-stranded while your RNA is single-stranded. Yung thymine mo, for DNA lang yan. For RNA, uracil. Ang sugar or pentose ng DNA, deoxyribose. Hydrogen lang yung nakadikit sa second carbon. While sa RNA, ribose. Hydroxyl group yung naka-attach sa second carbon. DNA only maintains the protein encoding information. Pero si RNA, siya mismo yung nagke-carry ng protein encoding information, di ba? Isa yan sa pag-uusapan natin later on. DNA cannot function as an enzyme while your RNA may function as an enzyme. Lastly, your DNA will persist, no? Maipapasa siya sa daughter DNA natin, but your RNA, short-lived siya. Once ma-perform na niya yung function niya, wala na po siya. So what are the different RNAs present in the body, specifically in the central dogma? We have 3, no? But in this image, 4 na po yung nilagay ko. So first, you have your messenger RNA. That's the meaning of M in mRNA, messenger. Then you have your tRNA, also known as transfer RNA. You have your rRNA or ribosomal RNA. And last is your small nuclear RNA. But for this discussion, we won't be talking about the snRNA. So let's talk about our RNAs one by one. First, your mRNA. It is a long straight chain of nucleotides which is usually the end product of transcription. Okay? Later on, we'll go to that, no? Basta tandaan, si mRNA ang produkto ng transcription. So what happens? Since DNA is too big and it cannot leave the nucleus, no? Di siya kasya sa nuclear pore, a short copy of the DNA will be made. And that copy is your messenger RNA. Okay? So, just like any other nucleic acid, it contains nitrogenous bases. And this is how it would look like. A single chain containing your nitrogenous bases. So our mRNA are important because, again, sabi natin, description ng RNA, they carry the information that codes for amino acids. At pag nagsama-sama ang amino acids, it will form a specific protein. Okay? So usually your mRNA is composed of 500 to 1,000 nucleotides long. And binabasa ang RNA natin, ang ating mRNA, in sequences of 3 bases. So magbabasa ng tatlong nitrogenous base, tapos tatlo ulit, and then tatlo ulit. And those combination of 3 bases is called codon. Okay? Codon. So usually, the mRNA always begins with the 3 base pairs of A, U, and G. A, U, and G. So that is your start codon. Other than that, itong tatlong base pairs na to, once it is read, it will also produce the amino acid methionine. So, all proteins made from your mRNA will always begin with methionine. And we also have stop codons. That's your uwa, uwag, and uga. Hindi na sila nagko-code for an amino acid. Kasi kapag nabasa sila, stop na kaagad yung production ng protein polypeptide natin. Okay? So, we'll go to discuss this later on during transcription. Next, we have your rRNA or ribosomal RNA. Similar to your mRNA, it's a single strand composed of 100 to 300 nucleotides long. Unlike your mRNA though, it appears globular in shape. So ito guys, itong paikot na to, gawa siya sa single-stranded RNA. Pero yung shape, pabilog. Okay? Globular in shape. Your rRNA are made inside the nucleolus. So, di ba? So, when we studied about cell anatomy in HAP, and even dito sa cytogen, we always say, no, nucleolus, important for protein synthesis. Eh, bakit siya important for protein synthesis, no? Parang hindi natin alam dati, but now we know. It's because they create your rRNA. And your rRNA is needed for translation later on. Pag-aralan natin yan. So, it is associated with proteins to form your ribosomes. And where can we find your ribosomes? In your rough endoplasmic reticulum, di ba? And we say always that ribosomes, no, help in protein synthesis as well. Then lastly, we have your tRNA, stands for transfer RNA. It is cloverleaf shape. Kita nyo naman, no? 1, 2, 3. Usually, ganun yung appearance ng cloverleaf, di ba? Also, it is a single-stranded molecule. And look at this. From 5', isang strand lang siya, papunta ng 3'. And at one end of the tRNA, it has an attachment for an amino acid. Okay? Sa dulo ng tRNA, may kadikit siyang amino acid. While the other end, kung ito yung amino acid end, the opposite end has 3 nucleotide bases, which is called the anticodon. So ito yung huling tatlong bases niya, no? C, G, G. Those last 3 opposite the amino acid attachment is what we call anticodon. So, anong importance nito? The anticodon should be complementary doon sa codon ng mRNA. Okay? So, later we'll go and discuss that in detail pa. So, putting them together, this is your single-stranded mRNA. Again, ito yung mismo yung kopya ng DNA natin, no? They are read in bases of 3s or in codons. Tatlo-tatlo ang basa sa kanila. Sino nagbabasa sa mRNA? Your ribosomal RNA. It is the rRNA that reads your messenger RNA. So, pag nabasa ni rRNA, let's say itong base pair na 1, 2, and 3, hahanap or tatawag si rRNA ng tRNA na may anticodon complementary doon sa nabasa natin. Okay? So, hahanap siya ng tRNA na magko-complement doon sa nabasa niya from mRNA. And once makatawag siya ng tRNA, yung tRNA na yon may nakasamang amino acid. So putting everything together, this is how it would look like. Once the mRNA escapes the nuclear pore and goes to the cytoplasm, it will look for your ribosomal RNA. So the ribosomal RNA will start to read the mRNA in bases of 3 called codons. So once it was able to read yung codon, so sabi niya nabasa niya AGU. So the ribosomal RNA will call for a tRNA that is complementary to your AGU, and that is UCA. So pupunta si tRNA na mayroong anticodon complementary doon sa mRNA. Pag nag-attach sila, bibitaw po yung amino acid. And those amino acids will form a chemical bond to produce the growing protein. So what is that first amino acid again? Always methionine. Kasi that is the start codon, AUG. And what will be the last? Either UA, UAA, UGA, UGA, or UAG, UAG. So, for transcription, it is the first step in gene replication. Okay? So, what happens? Because the DNA is too big to exit the nucleus, a small copy of it must be created. And this will exit the nucleus via the nuclear pore and go to the cytoplasm. Okay? So, ano nangyayari? We have a double-stranded DNA, no? We have a DNA here. Kailangan natin gumawa ng short copy of the DNA. So, it has to be split open by your RNA polymerase. Sa DNA synthesis, what opens the double-stranded DNA? Your helicase. But in transcription, what splits them open? Your RNA polymerase. This is the green structure that you see on your screen. Siya yung nagbukas doon sa ating double-stranded DNA. So, pag bumukas na, ma-expose yung mga nitrogenous base, di ba? Sa dalawang parent strand natin. One of those strands will be used as the template, and nucleotides complementary to the template strand will eventually form your mRNA, your messenger RNA. Kasi again, messenger RNA is the end product of transcription. So transcription basically occurs in 3 important steps, no? Initiation, elongation, and termination. Kanina sa DNA synthesis, sabi natin, si helicase will attach to the ORI protein of the DNA parent molecule. But for transcription, the RNA polymerase will look for the promoter region. Doon siya mag-i-initiate. Okay? So, initiation is the control point that determines anong genes ba yung kokopyahin, no? Anong parts ng DNA yung kokopyahin natin. So, usually, hindi lahat no, of the DNA, not the entire length, just short copies of the DNA. Eventually, once na bumukas na po, base pairs will be added to the template strand, and that is called elongation. And lastly, once matapos na, the RNA polymerase will encounter the terminator region, and that will send a signal to end transcription. Afterwards, the mRNA will exit the nuclear pore and will go to the cytoplasm and look for the ribosome. And that enters now into translation. So, in translation, the messenger RNA will be translated into proteins. Okay, so what happens? We have to first understand that each amino acid is formed by 3 nitrogenous bases. Later, I'll show you a slide showing that. And this translation process occurs in the cytoplasm, where the ribosome or the rRNA reads the messenger RNA in groups of 3 bases called codon. So again, yung DNA template strand mo, no, yun yung nag-produce ng messenger RNA. Si messenger RNA babasahin ni ribosomal RNA para babasahin niya in groups of 3 or in codons. And then, pag nabasa na niya, the ribosomal RNA will call for the transfer RNA containing the anticodon na swak dito sa binabasa nating codon. Once mag-attach, it will release the amino acid and will contribute to the growing protein. Yan. So, this is how it happens. This is your ribosomal RNA. It reads the base pairs in groups of 3. Once it is able to read it, it will call for a tRNA, no, that will attach doon sa ating mRNA. So again, the first one is always methionine. AUG, AUG. All right? So these are the different codons encoding your different amino acids. Again, your methionine is also the start codon, AUG, and you also have your stop codons, UA, UAG, and UGA. And eventually, once those amino acids will lengthen, it will become your protein. Okay? So, I guess that's it. That's your central dogma. All right? So, that's the end of my presentation. Hopefully, you were able to learn something. And good luck on your prelims. Shalom.